Thank you all for joining our webinar this afternoon on Maldi imaging reveals new insight into glycosylation and glycan distributions in cancerous tissues. My name is Nancy Wright-Ross, and I will be your host. If you should have any technical difficulties during the presentation, please use your chat box and communicate them with me. At the end of the presentation, we will have time for a brief question and answer session. We ask that you use the Q&A box during this time and select all participants so everyone can see the questions that are being asked. If we're unable to get to your question, during that time, you can contact our speaker offline and we will provide that information. I would now like to introduce our presenter for this webinar, Dr. Shannon Cornett. Dale Shannon Cornett, PhD, University of Georgia, 1993, joined Bruker in 1994 as applications scientist, and by 2002 was product manager of benchtop Maldi systems. He then moved to Vanderbilt University to become research assistant professor of biochemistry, working with Professor Richard Caprioli to develop new tools and methodologies in the then emerging field of imaging mass spectrometry. Shannon rejoined Bruker Daltonics in 2009 to support imaging products and now manages the MS imaging business in Americas and Asia Pacific. He continues to hold an appointment as adjunct research professor in biochemistry at Vanderbilt. At this time, I will pass this presentation over to our presenter, Dr. Cornett.
Thank you, Nancy, and thank you, everyone, for taking time out of your busy schedules to join today. The topic of the presentation is Maldi imaging and how it can be used to gain additional insights in many biological samples, but the specific examples of topic today is a relatively new field of using Maldi imaging to understand better glycosylation patterns and glycan distributions in cancerous tissues. So before we dive into the Maldi imaging application, I think it's important to take a few moments to kind of look at the current state-of-the-art traditional how things have been done. There are many of you already practicing various omics type strategies, be it proteomics, lipidomic, or metabolomic approaches to studying tissue and cell biology. And as illustrated here on the slide, all of these compounds, starting with cell line or tissue, all of these various omic strategies begins by taking those cells or the tissue and extracting, fractionating out the class of compounds you're specifically interested in. Of course, in the case of proteins, typically that's followed by an additional digestion step where that very complex protein solution is digested into an even more complex peptide mixture. And as you might imagine, given all of the complexity within the various numbers of proteins and compounds that are extracted from the biological media, this results in an extremely complex mixture, specifically giving rise from a mass spectrometry standpoint to a number of challenges for the MS analyzers. Specifically, as I mentioned, we're starting out with the extraction of several million or several, tens of thousands of proteins per cell. Once that's digested, then we're reduced to several million potential peptides, including post-translational modifications. From the lipidomic standpoint, there's tens of thousands of lipids possible. I think currently in the lipid maps database, curated and theoretical, there's about 40,000, a little more than 40,000 compounds in the database. From the metabolomic standpoint, there's tens of thousands of possible metabolites present in a number of databases that are available. Along with this just species complexity is the fact that they're present in various ranges of concentrations, particularly focusing on proteomics. It's been estimated there's about 10 orders of magnitude in concentration from the lowest to the most abundant proteins, so that certainly presents a challenge to the mass spectrometry from that dynamic range. And then ideally, in any of these experiments, we would like to end up with an identification of the compound, be able to point what it is, not just its intensity change, but what is it? And ideally, again, all this would be done in as fast as possible in real time during acquisition, if at all possible. So there's a number of various challenges that these approaches really present to the mass spectrometry. And, focusing a little bit more on the classical proteomics approach, there's a number of additional, just going beyond sample complexity, particularly as depending on what the particular application is on the left side, left column of things, from tissue biopsies to clinical and so on. We can be faced with a limited amount of sample. If we only have a few cells from a cell line or a few cancer cells in a tissue, then we certainly have limited sample capacity. On the flip side, we can have large numbers of samples, particularly if we're dealing with clinical samples. There could be very large sample cohorts. We can be dealing with quantitation, particularly when faced with such a range of abundance, compound abundance. And then, of course, selectivity. We want to be able to detect as much as possible, but certainly from an ionization standpoint, we may not be able to effectively and efficiently ionize every compound that is present. But nonetheless, there are solutions available, particularly from a metabolomic standpoint. Bruker has an extremely nice platform, starting with the Elute UPLC Impact 2 system, operating under the Metaboscape task software combo. And various number of databases there, as you can see, are available from the identification standpoint. The proteomics side, certainly, hopefully by now you may have learned that Bruker's newest introduction is the timsTOF Pro with PASIF. Is compatible with either Peaks or MaxQuant software, and it is operating pretty well, better than most any other system available. Tops the numbers of protein IDs from just a very small few nanograms of cell line digest. So there's two real good classical approaches here from the traditional omics standpoint, but looking a little forward, there's a couple of downsides if you think about it. First of all, the sample extraction process. It is a very efficient process for extracting a very wide range of chemical compounds, as you saw with the metabolites to lipids to proteins. But there's two limitations to homogenization extraction. The first is that tissues in particular, are comprised of heterogeneous cell networks. And so if you're taking a biopsy tissue, which probably will contain only a few % of the cells in that tissue are the cells of interest, then if you homogenize the tissue itself, then all of those other cells that are not interesting, at least at this point to you, are introducing then that homogenate. Those other cells are contributing a huge amount of chemical contamination. And additionally, that process of homogenization in the presence of all this other contamination, these signals and the compounds from the cells of interest are actually getting diluted out. Oftentimes, it may be below a limit of detection, again, coming back to the dynamic range issue. Now, we see in the literature often that this has been addressed in a few studies with laser capture microdissection. So you take sections of tissue and you extract from the tissue just the cells of interest. And that certainly produced some good results, but just the fact of LCM itself brings in some other limitations. How many sections do you have to process to get sufficient material for handling, for practical handling and the like? So it's not the panacea of solving the problem. And then the other factor there, extraction, in the process of homogenizing the tissue or throws away or discards any spatial information. So not only are you diluting out the signals from the cells of interest, but if you are able to detect molecular changes you're never really 100% sure that they're actually originating from the cells of interest in an homogeneous extracted solution. So with this kind of thing, we want to look for ways of going solution-free. And one of those ways is using Maldi imaging. So Maldi imaging, in a nutshell, is described as a label-free tool for producing distribution maps or images of compounds distributed throughout a thin section of tissue. As illustrated here, we start off on the left with a thin section of tissue on a microscope slide. We do some processing, and we'll discuss that in next slides. But we do some processing, and we collect Maldi mass spectra from various locations on the surface in such a way that any compound that we detect, we can map the intensity of that compound as a function of its location on the sample. So the result in a single measurement is shown on the right of the screen and illustrated as a panel of molecular images. Each color here each image represents a different compound that was detected. And the intensity of that color represents the intensity of the compound as it was in the mass spectrum. So if you look within an image, then the intensity as you go from right to left, in this example, it's a coronal brain section. But if you look at the intensity of that compound throughout that section, the intensity represents a good measure of its abundance in that section. So it's a relatively powerful tool of getting hundreds and sometimes thousands of molecular images from a single tissue section in a relatively short amount of time. And so that brings a number of advantages to the table. And if we just zoom in a little bit, you get a little bit better detail of this panel. So here I'm just showing 9 different molecules, their distribution throughout this rat brain section. And so because it is mass spectrometry technique, we do have the specificity of mass spectrometry. We we make the images and collect them without any form of labels. So it lends itself really nicely to untargeted discovery type measurements. It carries the widest range of compound detection of any mass spectrometry imaging technique. You can use, depending on the sample processing, we can map panels of metabolites, lipids, peptides, proteins, all the way from very small to relatively large compounds. Because we are collecting hundreds or thousands of these molecular pictures, we can explore within the data sets intermolecular relationships. We don't have to look at each peak, whether it's up or down. Now we can start comparing multitudes and combinations of peaks, what goes up as something else goes down, and any combination you can think of. We can use this data sets to explore different relationships. It is imaging, so it does integrate very well with other imaging modalities. If you have for example, if you have a fluorescent assay for a compound within the same model, as long as you can visually register your fluorescent assay image to the images you see here, we can overlay those in the software and get very nice contextual descriptions and illustrations of these ion images relative to the fluorescent or even immunohistochemistry images. The power of being able to do that, bringing in other images and overlaying them with Maldi images. Most commonly, this is done with histology images. But the real power in this is being able to associate and immediately see the molecular expression from the ion image related to the histological portion of the sample so that you can immediately see what types of cells are actually giving rise to the molecular signals. Now, moving a little bit forward, just to illustrate the workflow, Maldi imaging has only been around since the late 90s, so it's relatively new in the scheme of things. Very recently, it's really exploded in terms of application range, but it is a relatively simplistic workflow, straightforward, starting out with sample cohorts. This could be animal models, this could be clinical samples, any type you want. It could even be plant or inorganic. The focus here is on human tissues, but it could, you see applications for MALDI imaging in a variety of fields. So we start with our sample cohort. We section thin sections, mount them on a microscope slide. We apply MALDI matrix in the form of an aerosol to generate a very uniform thin film of matrix on the surface of the sample. And then we load that in a mass spectrometer, acquire the data, and then once we have the data, depending on whether it's a targeted, we're just wanting to see visualization of where distribution maps of certain compounds, drug studies, for example, we just enter in the molecular weight, we see immediately where that compound is distributed. If it's an untargeted study, as kind of illustrated here in this the image on the screen, we take the cohorts of samples. We either segment manually by histology, pulling out just those molecular signals from the cells of interest, or we can let software do that for us, and I'll talk a little bit more about that in the coming slides. So the real power of MALDI imaging is it gives access to a wide range of compounds, really only depending on how you process the sample. Starting as illustrated here, it hinges in this middle portion with the device here called the TM sprayer. We can process the sample slides in various baths. For example, if we're imaging proteins, we can dip the sample in a bath to remove metabolites and lipids and salts. That way it enhances our protein signals. But there are a number of treatments that we can do to the sample itself before applying matrix, depending on the class of compound we're looking for. And then the matrix that we spray with the TM sprayer really helps us focus in on a specific class of compounds. There's matrices that are better suited for metabolites, better suited for lipids, better suited for proteins. But the important thing is here, the one technique can access all of these with just some minor variations in sample processing. So the real component of MALDI imaging that lends its power is being able to take these, in many cases, hundreds of thousands of spectra and render those data into useful information. And what Bruker offers is Skills Lab software. This is produced by Skills, and it reads in Bruker data, and it offers a complete statistical suite of tools that we can apply to our data. Just a subset of those toolkit is shown here on the screen. We can do co-localization. If we know a region of interest, a tumor, for example, we can ask the Skills Lab software to pull out and extract all compounds that are most abundant in the tumor region or most abundant out of the tumor region. We can look at ROC curves. How good is a particular ion as a classifier for two different regions of the sample? Principal components. If we have multiple regions that we want to look and differentiate, we can use principal components and the like. So there's an entire a cadre of statistical tools that are available and ready to apply for our MALDI imaging data. So detecting more, everyone always wants to detect more from a particular sample. So we can detect proteins, we can detect lipids, but extending beyond that, certainly subsets are more readily detected than others, and also kind of dependent on whether we're looking at fresh samples or formalin-fixed samples. We have different restrictions, different options available to us. One of the ways of broadening the scope of what can be detected in MALDI imaging is using enzymes or derivatization agents. These are actually applied directly to the sample. And, in the case of derivatization agents, there are a number of publications that have shown that small metabolite molecules, primary amines, for example, may not ionize very well directly from MALDI, but if you derivatize the amine functionality with a certain compound, it makes them much more amenable to ionization. Similarly, enzymes like trypsin can be applied to tissue. Trypsin will digest the large, more insoluble proteins into more soluble peptides, making them a little bit more readily detected. In the case of formalin fixed, we're pretty much required at this stage of development to use trypsin to break up that chemical cross-linking network. The action of formalin on tissue is to cross-link the amine functionalities and amino acid residues. And so to liberate something that can be detected from that heavily cross-linked network, trypsin is a really good enzyme to do the digestion. So from FFPE samples, we're primarily restricted to looking at tryptic peptides. But one of the topics, I guess, for the rest of the presentation is the fact here that there's a small but growing number of groups that are looking at an enzyme like P-engase F as a way to get more information for P-engase F. It's a way of getting access to glycosylation patterns and then correlating the glycosylations with pathology. And so just kind of to illustrate the potential growth of Maldi in the whole world of imaging, mass spectrometry imaging in particular, you can see here just looking at Maldi and compared to other ionization sources. Maldi is not the only ionization source that's compatible with mass spectrometry imaging, but as you can see here, looking at the number of publications by each year, you can see Maldi dominates the amount of scientific literature from the ionization standpoint. And there's a number of reasons for that. Maldi being laser-based, pretty much you can just turn on the instrument and it starts its acquisition. There's very little tuning involved. Maldi itself is a very robust, reproducible technique, and it's particularly since we're dealing with tissues, which is a very complex but also a very dirty and biological environment. Salts and everything, fats floating around. Maldi is a very robust technique. It's large, has a high insensitivity to contamination. And so when you look at all the breadth of what's in the literature, there's largely dominant by clinical research, looking at clinical samples, trying to either uncover new potential biomarkers for various diseases, or looking at taking samples and classifying, making classifications of various cancers. In many cases, histology, pathology may be indeterminate diagnosis, looking at ways of possibly using growing mass spectrometry imaging into a way that we can augment that kind of information from the molecular signatures and molecular level. So that brings us to kind of the meat of the rest of the presentation, focusing on this small subset of Maldi imaging, particularly with glycans. Glycosylation proteins, quite heavily important in cancer development. About 80% of proteins on a cell surface are glycosylated. Glycosylation, specifically, the entity, the glycan entities, plays a key role in cell signaling. And certainly it's well known that glycosylation are altered during tumorigenesis. And so about half of the proteins are glycosylated in some form. Most of those do reach the plasma proteome. So there's lots of things floating around. Ideally, plasma, blood, fluids are much less invasive testing environment. So eventually we'd like to identify things that could be markers for minimally invasive assays. And then the majority of FDA-approved markers are in fact glycoproteins or glycan antigens. So it's clear that glycans and the identity of glycans certainly plays a significant role in tumorigenesis and so on. We can look at glycans, glycoproteins, glycosylations at three different layers of granularity. Starting off here with the parent glycoprotein, glycoproteome, we can look at those intact species for just a straightforward profiling type measurement. We can digest at the bottom. We can digest this glycoprotein into glycopeptides and then make a mass spectrometry analysis on the glycopeptide mixtures, identifying glycans, identifying the specific peptide backbone, doing a complete profile in that sense from the peptide level. But then we can also release the glycans, just knock them off the protein backbone, and then in solution make identification and structure elucidation. We can do profiles, what types, what are the ranges of glycan species at each amino acid, what's the distribution and the like. But I'd like to introduce here a third alternate with the released glycans, and that is glycan imaging, using Maldi imaging to map glycans. And so we can kind of look at this. It's a little bit of a simplification in the sense that mammalian glycans are composed primarily of 10 polysaccharide units. There can be some modifications of those, but in general, there's not an infinite number of combinations. With this just 10 building blocks, we can kind of get a more discrete, more manageable subset of glycans to deal with from the complexity standpoint. I mentioned earlier the enzyme P-engase F, is shown here. It liberates N-linked glycans. There are O-linked glycans, but unfortunately at this point of development, there's no real equivalent enzyme that cleaves the breadth of O-linked glycans as effectively as the N-links, although a number of groups are working on developing these enzymes. But for right now, the majority of the work that's been done with Maldi imaging has been restricted to P-engase F measurements. And so just looking at this, the workflow, it's a pretty much very similar to a trypsin digest. So be it trypsin or be it P-engase F, we start with our tissue section, be it fresh frozen or FFPE. We wash and we process, and then we apply the enzyme. In this case, P-engase F. In the trypsin case, we would apply trypsin. We digest, we incubate, and then apply our Maldi matrix to the sample, and then acquire our Maldi image. As I mentioned, the building blocks, the limited number of building blocks makes for a less complex spectra than if you were to apply trypsin. You can imagine if you start with 10,000 proteins, apply trypsin, we pretty much will see in the mass spectrum a peak at every mass, and if you can resolve those even further, we can get dozens of peaks at every nominal mass. But here you can see kind of the less complex spectra. One of the nice things, and it is shown here in the workflow using FFPE tissue, one of the nice reasons for using FFPE tissue specifically for the P-engase F measurements is that we don't have to break up the protein cross-link network. That helps keep our baseline spectra here free of peptides. So most what we detect here are just simply the released glycans. Now, in an imaging experiment, certainly we're looking for distributions of any of these species, and I show 5 here, taken from a colorectal carcinoma section. And you can see 5 different glycan mapped, certainly in completely different locations within the biopsy. So you can see that just like proteins, just like lipids, the glycan species have a regiospecificity of localizing to various regions of the sample. A number of researchers across the world are looking at glycans. The majority of the names here are involved in imaging measurements in some form or another, and many are actually involved in various other extraction type approaches as well. But there are a number of people out there. It's a relatively small subset of the Maldi imaging world, but certainly it is a growing community. So looking at some more data, colorectal carcinoma here again, the same sample as the previous slide. The panel you see here is an extraction. Each panel is a distinct glycan species. And so you can see the breadth, not only of the breadth of the number of species that are detected, but also their distribution is quite a diverse subset. Zooming in a little further on just a few of them, again, you can see things that are primarily found in tumor, primarily found in the crypt, primarily found in various other regions of the sample. We can take this and illustrate this diversity a little more. We submit all of these data to Skills Lab software for segmentation. And the segmentation looks at each spectrum. So remember, each pixel in this image, in the images you see here in the center of the screen, each pixel is one peak out of a complete mass spectrum, a complete fingerprint of all the glycans at that specific location. And so we can take those spectra, submit them to a clustering algorithm, which looks at degrees of similarity, and mapping out this each region of the sample as the color here, not the color here in this particular segmentation map is not a specific M over Z, but it's a specific degree of similarity. So you can see how the similarity of those molecular signatures mimics quite well the histology of the sample. And just illustrating here, these segmentation maps were generated from looking at the patterns of 102 different glycans that were detected in the data set. We can look at and detect things that are regional specific, but also we can see that different types of glycans may prefer different regions of the sample. So for example, you see here in the colon tumor H&E biopsy in panel A. In panel B, we've got an N-glycan elevated in the stroma region. Panel C, we've got a high mannose glycan elevated in the tumor. We also have glycan elevated in the stroma, panel D. N-glycan elevated in the crypt, panel E. And then panel F, a glycan specific to the mucinous tumor region. So there's some degree of specificity related to the inherent makeup of the glycan moiety, and that's kind of seen and illustrated a little more detail here, looking at just high mannose glycans. Same biopsy, same sample. We see that a number of high mannose glycans are specific to the tumor region, and that's the high mannose. The presence of high number of mannose is illustrative of the disruption in the synthesis pathway of the glycans, and so it's whether or not this is a cause or an effect of being in the tumor is not known at this point, but certainly it's indicative of some type of disruption in the synthesis pathway. Looking at another example here, prostate tumor. Two different Gleason scores, a 6, a 7, and a 9. Again, segmentation maps, individual glycan maps, illustrating certain glycan moieties preferentially abundant in the tumor region, preferentially abundant in the stroma region. And then again, based on the segmentation of 80 different detected species, you can see a nice agreement with the general histology, just giving support to the idea that the glycosylation patterns are very relevant to the tumorigenesis. So focusing now on a little bit more of a smaller biopsy, but more patients, tissue microarrays are a very nice way of getting access to large numbers of patient cohorts in a one single measurement rather than multiple larger biopsies. So you can see here we've got one in this, the H&E section here in the top left. We have one normal core and 2 tumor cores from 16 different patients in hepatocellular cancer. Looking at 2 different glycans in the lower panels, you can see one species is elevated primarily in tumor. There's some presence in the non-tumorous regions, but it has a preferential abundance in the tumor regions. Similarly, then panel D, one glycan species elevated primarily in the normal non-tumorous regions. Again, just as the other one, there are some examples presenting in the tumor regions, but primarily the abundance is in the predominantly normal tissue. And just in the top right, just an overlay of these 2 images to kind of pull the colors all together. One of the goals in terms of eventually being able to take this technique into something of clinical relevance is to be able to look at large numbers of samples, make quick classifications based on single biopsy images against large libraries. And one way of generating these larger libraries is looking at multi-tumor, multi-tissue TMAs. And illustrated here, we've got various tumor TMAs from a number of different originating organs, all in a FFPE block, 178 cores total. We're also looking at native and amidated salic acids. Salic acids are notoriously fragile in the ionization process, and so if we can derivatize those, it's known that it stabilizes them and makes them a bit more detectable. But just as a one little quick data slide from one of these TMAs shown here, you can see at the top the distribution of organs, cancer types. The bottom illustrates quite nicely that, yes, we do get specificity from the different organs with the various glycans, which produces a large number of distribution maps, which helps us then to have a basis for generating potential libraries in a very short order from a large number of patients. And so with that, I would like to summarize and just to say that, I hope I've shown you that Maldi imaging is a very powerful spatial complement to traditional omics strategies. And its powers is derived from the fact that it provides a regional and cellular specific molecular distribution capable of differentiating various disease states, various regions of the sample. And along with the fact that proteins, lipids, and metabolites present unique distributions from a regional specificity, glycosylation patterns fall in line. They differentiate cancer cells from healthy cells quite readily. Glycosylation maps from various types of cancers clearly correlate with organ and cancer. And then in the longer term, relating these specific glycans to a regional origin within various biopsies can only improve our understanding of disease mechanisms. And so for further reading, if you're interested, this is just a selection of some literature examples of glycan imaging. Certainly, there's a number been published in the last recent years. And with that, again, I thank you for taking time out of your busy schedule and would be happy to answer any questions.
Thank you, Dr. Cornett, for the great presentation and data. That was very, very informative. Yes, we're now going to open this up for some Q&A. I just want to remind everyone that please use your box and direct the questions to all participants so everyone can see the questions that are being asked. And you had a couple already coming through, so these might have been already answered in your presentation, but I'll go ahead and ask them anyway. First question came in earlier. Can you elaborate more on how Maldi imaging is used to map different compound classes?
Yeah, I think it was partially addressed in the sample prep slide, but the compound class that you want to go for, be it lipid, be it peptide, protein, metabolite, drug, and then even glycans, really it hinges on, each of those classes are made accessible by various processing steps of the sample. So we can either process and treat the sample, as I mentioned in the slide for proteins. If we're looking to image proteins, we can wash away the lower molecular weight contamination, if you will, to improve our specificity and sensitivity for protein ionization. And then also the matrix that we use also determines large part our sensitivity and what specific proteins we see.
Okay, good. Another question here. Do you see Maldi imaging applied more towards targeted or untargeted studies?
Well, I mentioned both, and targeted studies, at least at this stage of the technique development. Targeted studies are primarily employed by pharmaceutical and the drug discovery pipeline, where you're dosing your animal models and you're looking for known compounds, known metabolites, just to see where they're distributed. Probably the bulk of Maldi imaging publications and studies are revolving around untargeted discovery. And I think, eventually the two will come together once we use Maldi imaging to identify, reliable markers for whatever disease or state you're looking for. Then those markers then would turn into a targeted assay for later on.
Okay. Another one. You mentioned that you can use fresh, frozen, or FFPE tissues for Maldi imaging. Is there a preference?
I think it's generally accepted within the field that fresh, frozen samples always give the best signals.
Okay.
Unfortunately, when you're dealing with clinical samples, that's not always an option, since FFP is a standard processing in clinical realm. But understand that FFPE can be used. There are protocols, but it does involve those extra bits of processing from the applying the trypsin and so on to liberate things from that cross-link network.
Okay, someone else here says that they have read several papers that describe sublimation for applying the matrix. How is this different?
A number of groups are using sublimation, are applying the matrix by subliming directly onto the sample. That's a bit different than what I described. We use an aerosol. It's essentially a pneumatic nebulizer, a micro airbrush, if you will, that's on an XY stage. And so it is very reproducible, very reliable. Sublimation is, usually most groups are using their own home-built system, so they have their own protocols, and it may not translate very well to other systems. But the big difference is that sublimation is completely solvent-free. We use an aerosol, which the aerosol droplets hit the sample, and for that brief amount of time before the solvent evaporates, there's some extraction going on. And, various analyte classes require different stages of extraction. Proteins like to be heavier amounts of solvent, so we generally have a heavier aerosol for protein imaging. Solvent-free technique like sublimation, there's very little extraction going on. It works quite well for lipid imaging, but if you want to use sublimation as a technique for accessing other classes of compounds that need to be extracted, then there are ways of taking the sublimed sample, putting it in a humidity chamber, and using the humidified environment to do the extraction. So there's a little bit more complexity to it, but it is used by a number of groups.
And you may have answered this in your earlier talk, but I'm not sure. How were the glycans identified?
No, I did not. The identity and the mapping up there is based on a couple of factors. First is that wherever possible, they're identified by MSMS. Now, MSMS can only take you so far with identifying degrees of branching. In other cases where they can't be identified directly by MSMS, then it's either drawn as a nebulous. We've got a branch, but it could be on the left fork or the right fork. We can't determine that by MSMS, or it's based on the assignment of a structure is based on just prior knowledge of working with the sample.
Okay, and this question came up a couple of times. Would I be able to combine results from your system with a collaborator who uses a different system?
For the most part, you can by way of the Skills Lab software. So I mentioned that the Skills Lab software has a complete suite of statistical tools, but it also can be used for just basic visualization. And there is a version of the Skills Lab that is compatible with the open access IMZML format that is kind of the open access format for mass spectrometry imaging data. And so using that multi-vendor support version of the Skills Lab software, you can bring together multiple different sourced data sets.
Okay, we're a few more questions here. We're getting close to the end. Is there any way to identify the original glycoproteins from which the glycans were removed?
That has not really been completely done yet. I know there are some groups that are working with doing the p-engase digestion, washing off the matrix, and then following that with a trypsin digestion. I remember usually it's FFPE samples. And so that gives access to peptide maps, and just, the starting point is to look at regions where certain peptides are most abundant that are the same regions where certain glycans are most abundant, and trying some fashion to correlate that way. But at this point, it's just pure correlation, pure matching. There is no direct relationship been discovered between the glycan maps and trypsin maps.
Okay, a couple more questions and we'll have to close out. Can you image glycoproteins directly?
Okay. That's kind of related, I guess, to the earlier question. To my knowledge, there have been no studies of intact protein imaging that have clearly identified glycoproteins. So it's not to be said that it hasn't been done. It's just, to my knowledge, I'm not aware of something in the literature or some unpublished data of that nature to claim the direct mapping of glycoproteins.
Okay. What is the highest resolution that you can obtain?
Okay. Well, since we're talking imaging, I'm assuming we're talking, the question would be image resolution as opposed to mass spectrometer resolution. From the image resolution standpoint, we generally think of it in terms of what can you do on a routine basis. Quite often, you will find, and the natural inclination of everyone is to always want to push the limit. What's the highest resolution you can do? But in fact, because each pixel is a mass spectrum, it all boils down to, do you need to acquire 100,000 spectra, or can you answer your question by acquiring 50,000 or 20,000 spectra? The fewer the spectra means the lower the spatial resolution, meaning the larger the pixel. In many cases, you'll see in the published literature, the pixel resolution is on the order of 50 to 100 microns, which is certainly multiple cell clusters if you look at the average mammalian cell of 10 microns. So there are a number of things that you can do to avoid the highest spatial resolution. In fact, your biological question may not require it. But from a routine perspective, we usually go to about 20 microns. Again, it comes down to a number of factors, most of which involves your extraction, your aerosol. The sublimation solvent-free does give you access to potentially smaller pixel size, but most compounds require some degree of extraction, which means some amount of solvent, which is one of the ultimate determiners of your spatial resolution.
Okay, we only have time for one more question. How many different glycans do you detect?
There are publications showing anywhere from 100 to in the 170 to 200 range is probably the most unique species in a data set. As I mentioned, there's some platforms or MRMS platforms which have a higher pressure ion source. Turns out that higher pressure, it's not atmospheric pressure, it's not vacuum, it's intermediate pressure. Turns out that intermediate pressure regime is really good at stabilizing the sialylated glycans. So we generally, in the MRMS systems, we'll generally see a few more species than we do with the high vacuum systems, just the difference between the silated species that fragment in the vacuum systems as opposed to the MRMS systems. But in my conversations, the audience probably noticed that there are a number of contributions from Professor Richard Drake's lab at Medical University of South Carolina. He's been one of the pioneers in this whole glycan imaging technique, and personal discussions with him is that they're close to being able to equalize the number of species with the observed in the vacuum systems as opposed to the intermediate pressure systems. So there's good news in that regard.
Wonderful. Well, there was a lot of information that was covered, and I'm sorry that we weren't able to get to everyone's questions, but if you do have, you do want to contact Shannon with a wealth of information here, he can be reached at email Shannon, SHANNON, .Cornett, CORNETT, @Bruker.com. I'm sure he'll be happy to answer any of the questions that we weren't able to get to today. And we want to thank everyone for joining our webinar this afternoon. Also, if you want to visit our website, a lot of good information is on the site at www.bruker.com. Thank you again, and have a great afternoon and a great weekend.